Thank you for that kind introduction, and thank you for the opportunity to present our preliminary analyses of the impact of pharmacogenetic variation on Bictegravir and Dolutegravir concentrations in individuals with HIV and tuberculosis. I'm presenting this on behalf of my co-investigators. So Bictegravir and Dolutegravir are included on most international guidelines in the first-line treatment of HIV. These drugs are metabolized by UGT and CYP3A and are substrates for several transporter enzymes. So genetic variability in drug metabolizing enzymes and transporters may impact the pharmacokinetics and drug exposures of Bictegravir and Dolutegravir. So this pharmacogenetic analysis was nested within the INSIGHT trial, which is a phase 2b randomized controlled trial to assess the efficacy, safety, and PK of twice-daily Bictegravir in adults with HIV and tuberculosis receiving a rifampicin-based TB treatment regimen. So we randomized individuals with HIV and tuberculosis to receive either twice-daily Bictegravir or Dolutegravir as part of TLD during tuberculosis co-treatment, and then once daily thereafter. We followed individuals up for 48 weeks, and the primary endpoint for the was viral suppression to less than 50 copies per ml at week 24. We enrolled 122 individuals in the study, 80 in the Bictegravir arm and 42 in the Dolutegravir arm. So in terms of the baseline demographics of these participants, the median age was 35, 35% were female, 100% Black African ethnicity. This was a relatively sick cohort with advanced HIV disease, 42% with the HIV viral load at baseline more than 100,000, 60% with CD4 less than 200, and about 25% of CD4 less than between 50 and 100. So viral suppression in the study nevertheless was robust, and at the primary endpoint it was 95% in both arms. This viral suppression has been maintained to week 48 with 95% in the Bictegravir arm and 93% in the Dolutegravir arm. So we did semi-intensive PK sampling in the Bictegravir, the intervention arm of the study, and sparse sampling within the Dolutegravir arm. So for Dolutegravir, we measured pre-dose concentrations at week 8, 24, and 48. And for Bictegravir, we did semi-intensive sampling in about half of the participants, 43 of them, at week 4 and 12 during TB treatment, and then at week 32 after TB treatment completion. But everyone had at least a pre-dose or some sparse sampling done. So this is just a flow chart of the individuals that we included in the genetic analysis that had both PK and genetic data. So if you look just at the BIC, show this pointer. All right, so just at the BIC, those that had the pre-dose, there was at least of the 80, 78, 77, and 76. And then we did non-compartmental analysis of the semi-intensive samples and calculated the AUC and C-trough. And then with Dolutegravir, we had pre-dose samples in almost all of the individuals. So just to remind you about the PK data for the Bictegravir, just to show that, we expected the BIC drug levels to be significantly reduced during rifampicin co-treatment, and I can't see it very well, but the Bictegravir concentrations during TB treatment, you can see even dosed twice daily with TB treatment were reduced to 0.397 compared to 2.29 at once daily without RIF. So in terms of the genetic study methodology, DNA was extracted from buffy coat samples that were collected at baseline in our trial and prepared for GWAS analysis, and which was performed on the Affrimetrix gene titan using Precision Medicine Diversity Array Plus. Data analysis was done in R, and the Mann-Whitney U tests were applied to compare drug levels between genotype groups. So I apologize for the busyness of the slide, but you can see from the previous slide that, based on the GWAS, there were about 850,000 SNPs that were included. So we decided to do a hypothesis-driven approach. We had a small sample size in this trial. And so we focused on just the genes that were involved in a drug metabolism or transport, and I just listed a few of them here. And the way we did this was because we had done multiple hypothesis testing, we had to correct for these multiple tests, and we used Bonferroni correction to do so. So just as one example, for ABC, for the ABC gene, there were 147 polymorphisms. Get this to move. All right, it's pointing at ABCB1, so I'll just use that example. So for here, there's N equals 27, so we had to adjust for the 27 polymorphisms for that specific gene. And for that, one of those SNPs to be significant, they had to be significant at 1.85 times 10 to the minus 3. And we did that across the board. So just focusing on the BIC that I showed you in the flow chart, we analyzed a large number of SNPs. And I'm just going to focus on one or two that were of the highest significance. So I've just circled the first two that I'm going to show you. So this one is on a gene level. We also did the analysis on an allelic level, but on the gene level, this was one of the SNPs that showed significance in the ABCG2. That was one of the genes, RS100-32109. And you can see, based on the drug concentrations, this was pre-dose concentrations of BIC. Individuals that had the CC genotype had an association of a higher drug concentration compared to the TT. So they had about a 3.38-fold higher concentrations than compared to those in the last group with the TT genotype. And I list as well the allele frequencies. So this has the C allele, which is the reference allele in this, or the major allele is 0.9 in the African population. Similarly, for this gene UGT2B10, having a TT genotype was associated with a 0.4-fold higher drug concentrations in comparison to the TG. And then we also did this on an allelic level, where we looked at each of the alleles. So having a T allele specifically we actually did this across all of the different time points, etc. Because, as I said, we still looking at this is an exploratory analysis at this point, and across the board at all of the different time points, having a T allele for the CYP2C8 gene was associated with a higher drug concentration compared to the T allele. And some of these SNPs may not be directly involved with the metabolism, but they also in linkage disequilibrium or linked to other alleles, and we're looking at that as well. I'm not going to go through all of these. There were quite a few. So with Dolutegravir, this is one of the ones with one of the quite high significance. I just highlighted this solute carrier, RS7623925, and this is again on an allelic level, where the having individuals with the A allele were associated with higher drug concentrations than those with the G allele, with a 0.28-fold reduction. So we can see that there was significant genetic variability in drug metabolizing and transporter enzymes that was seen in this African cohort. And genetic variability did result in significant differences in drug concentrations of Bictegravir and Dolutegravir. But we're not quite clear on what the clinical relevance of these differences in drug concentrations would be. I mean, the viral suppression in this trial was, in this cohort, was relatively high. We didn't have any failures in the study, but there may be implications for lower BIC concentrations, particularly in this trial with rifampicin reducing Bictegravir and Dolutegravir concentrations due to the interaction. These were sick individuals with other opportunistic infections. You can have other drug-drug interactions. Imperfect adherence, certainly we had that in our trial, and we certainly have that in our large ART program in South Africa. So, and then, when you add to the mix things like drug holidays, which I think our participants in the programs take, regardless of whether we ask them to take drug holidays or not. These genetic variants and differences in drug exposure could become significant. So these results need further validation, and we're going to be modeling the Bictegravir data or PK data in the trial. And so these models, we hope, will exclude other sources of variability. Excuse me, sorry. So I'd just like to acknowledge all our study participants and their families, our inside study team. And genetics is definitely not my comfort zone or area of expertise. So the nice analysis was done by the team at the University of KwaZulu-Natal, led by Professor Veron Ramsaran and his postdoc, Dr. Anmol Gokul. The Dr. Telehun Asena is our biostatistician. I'd like to thank my co-PIs on the INSIGHT trial, Professor Kelly Dooley and Kogi Naidoo. Our team at the University of Cape Town Pharmacology Lab, Roland Bassman and Paolo Denti. Our funders, the NIH, the South African Medical Research Council, and Gilead Sciences. Thank you.